essentially the product that we can make there has an incredibly low toxicity profile. It is incredibly clean. It's actually agnostic to length, so we can make much longer molecules than typically people can make with single-stranded DNA.
Hello, you're watching Proactive. I'm joined by Amy Walker, the CEO of 4basebio. Amy, very good to meet with you. You took over as CEO earlier this year. For investors who don't know 4basebio yet, what problem does synthetic DNA solve that plasmid DNA can't? And why is that shift gaining momentum now?
Thank you, Stephen. I think by definition, 4basebio is a company that makes DNA, and the way that we make DNA is fairly unique because instead of using conventional fermentation that is typical of plasmid production, we actually use enzymes to amplify our DNA. And so what this means is a much cleaner, safer, cost-effective, and faster type of DNA that we can deliver to our partners, be that biopharma, be that biotech companies, or be that some of our CDMO partners. I think with respect to where we can feed that DNA into, really DNA is used as a building block in all of cell and gene therapy and more broadly in vaccine applications and gene editing applications. And so essentially what 4basebio can do is make these application-specific molecules that serve a benefit for whatever downstream application that they're using. I think if we think more broadly about that plasmid DNA market, which is of course the incumbent technology in our space, depending on where you look, that market is worth upwards of $3 billion. And so there's a really sizable market that we can penetrate into with really the delivery of our synthetic alternative to that product. The last thing I would mention here is that we have our GMP certification from the MHRA, and essentially what that allows us to do, that is the UK regulator, and that allows us to make material that is suitable for dosing in patients and in humans through clinical trials and beyond. And so I'm very happy to say that some of our partners have taken this into clinical trials. The furthest advanced of that is a phase 2 clinical trial.
Since stepping into the role, you've signed a clinical supply agreement worth a seven-figure revenue stream, also launched a new ssDNA product line. How do these fit into the commercial growth strategy you're driving forward, Amy?
Yeah, I'll maybe start with the latter part of that question, Stephen. So we did recently launch our single-stranded DNA offering. And I mentioned previously that we really want to disrupt anywhere that plasmid DNA can be used. So this particular molecule, this single-stranded molecule, is ideally suited for gene editing applications where people are wanting to integrate or correct a gene using a gene editing technology like a CRISPR or a base editor, for example. And essentially the product that we can make there has an incredibly low toxicity profile. It is incredibly clean. It's actually agnostic to length, so we can make much longer molecules than typically people can make with single-stranded DNA, and that's really helpful for some of these strategies. You mentioned as well one of our recent announcements about our phase 2 clinical trial with one of our partners. That's quite an exciting one for us because typically, and when we first started to kind of commercialize our DNA offering, the expectation is that we would have to start in very early stages of development, pick up those customers and those partners at preclinical stage so that they could get comfortable and familiar with our technology, and then we would remain sticky and see them through into GLP tox, IND enabling studies, and ultimately into the clinic. What we're actually seeing, and it's very exciting actually, is that people are recognizing that actually they can make a switch to a synthetic DNA alternative later in development. And that's what's most exciting about this client. So this partner, for example, had already run some phase 1 clinical trials on their oncology asset. They were using plasmid. And they had a couple of frustrations with using plasmid with respect to turnaround time, with respect to product quality. And so they engaged with the regulator and were allowed to make this switch at a later stage of development. And so we hope to see more of that as we grow our commercial pipeline. I think from a company growth and a revenue perspective, this is incredibly important because the batch sizes are much larger, the product quality is much more significant, and as a result of that, the PO values associated with such opportunities are substantially higher.
You just added Jeff Coller, a leading mRNA biology expert, to your strategic advisory board. What gap does his appointment fill, and how will his input shape the platform going forward?
So we're absolutely delighted to be able to appoint Jeff onto our strategic advisory board. Jeff and I have worked together for some time across a number of different programs, and he really is a believer in cell-free DNA. I think he's one of those kind of key opinion leaders that recognizes the benefits and how we can solve problems that are typically associated with plasmid DNA, whether that is for, a non-communicable disease, which is incredibly large patient populations like a pandemic response or a seasonal flu vaccine. But also, and of critical importance, is actually the other end of that scale when we think about personalized cancer vaccines or N of one medicines. Really, that's where synthetic DNA can play and perform incredibly well because we can tailor our batch sizes depending on the number of patients that need to be dosed. And so, from a turnaround perspective, which is of utmost importance in these very critically ill patients, that's where he thinks we can be particularly disruptive. I mean, Jeff is an incredibly well-known industry leader in the US, and particularly with some of the headwinds we have faced as an industry with respect to the administration changes and some of the anti-mRNA sentiment in the US as well. Jeff is really standing up against that and thinks synthetic can play a part of that. He is incredibly polished with respect to how he engages in boards, and we really think he can help us drive that strategy and increase our visibility as a company, having him, with that vote of confidence.
4basebio also moving into a new state-of-the-art manufacturing facility in Cambridge. What does that transition unlock in terms of capacity and capability?
Yeah, we're very excited about our new building. So I'd like to kind of tell you a bit of the lay of the land there. So currently we have 3 sites in Cambridge, UK. One is our GMP certified facility. So that's the one that the regulator has come and visited. We have 7 clean rooms there. That's staying, and we will continue to operate there alongside our infrastructure that supports that from a QA and a QC perspective. What the new building really unlocks is much higher capacity for those earlier stages of development. So as we onboard customers, we make some research grade material for them. So it will significantly improve the throughput there. In addition to that, and very excitingly, we are going to be building out some custom suites there that are suitable for this N of one medicine that I spoke about. Being able to manufacture these personalized batches in a very high throughput way. And so that will augment the already established capacity that we have in our GMP suites as well. The rest of the building will house our R&D facilities, all of the platform development and that innovation that we continue to promote at 4basebio. I mean, naturally, a technology like ours is governed very, very strongly with this IP moat, and we continue to foster that in everything we do here.
Key milestones investors should watch out for the rest of the year as you push into this next commercial phase, Amy?
Yeah, I mean, I think it's commercial traction, right? So it's more of these GMP deals that we want to announce. Those are the really exciting ones. Those are the ones that are going to get into patients quicker. And, candidly, those are the ones that are associated with the highest revenue stream. So I'm incredibly encouraged with the significant pipeline growth that we have had since the appointment of our new Chief Commercial Officer and the build out of her team. And we're really excited about all of the GMP opportunities that we can see coming through. I think the last part that I would mention there is the kind of multi-modality approach that we deploy at 4basebio. We don't want to be kind of pigeonholed into one type of therapy, be that vaccines or be that cell and gene, for example. Actually, we want a really diversified portfolio such that we can deploy our technology in whatever is cool in biotech at that time, whether that might be in vivo CAR T approaches, new gene editing, or the seasonal flu vaccines that we continue to have. So I think we should anticipate significant revenue growth off the back of these contracts, and we feel very excited about that.
Amy, I hope you'll keep us updated with your progress. Thank you very much for the introduction today.
Thank you.
Amy Walker, the CEO of 4basebio.